Donald Trump recently made a claim that violent offenders who are immigrants have certain bad genes that make them commit more crimes than others in the country. Trump's claim is justifying the deportation of immigrants without due process due to assumptions about their morals based on what he claims is genetics. But in reality, it's just a judgment based on differences in the way they look and the place they came from. The truth is, biology doesn't care about these things, but to understand this, we must zoom in to our very first cell as a human being. We all began as a single cell. This cell contains 46 chromosomes, or a complete unique set of your human genome. However, inside present-day you, all trillions of cells in your body have the exact same chromosomes. How did they get there? That one cell duplicated in a process called mitosis, but before it divided, it needed to make sure that it had an extra set of DNA, one for each of the two cells it was becoming. During what we call the synthesis phase, our DNA is split down the middle. DNA is comprised of 4 bases, which code instructions for your body: A, T, C, and G, representing adenine, thymine, cytosine, and guanine. They bond to each other complementarily, with A always bonding to T and C always bonding to G. When DNA is split to be replicated, these form a duplicate strand. Now, your one little cell has 2 copies of genome stored in the form of replicated chromosomes, and it's ready to duplicate. This duplication happens through the process of mitosis. In mitosis, one cell becomes 2 through a series of phases. Chromosomes condense, and proteins called spindle fibers attach to them at the center. The spindle fibers align the chromosomes in a line at the center of the cell during metaphase, and pull them apart in 2 sets of unreplicated chromosomes, isolating the 2 copies of the genome. The membranes of the nucleus and cell split and form into 2 new cells, and now there are 2 cells with identical genetic information. Our one embryonic cell has now become 2, and through this process, we can see how it will keep multiplying to become 4 and 8 and 16 until it becomes the trillions in our fully formed bodies. But how do these identical cells become different, like our hair cells versus our blood cells or our skin? It's through a process called cell differentiation. But first, back to chromosomes. Different segments of DNA in a chromosome are called genes. Each gene contains instructions for making different proteins, which are responsible for the shape, function, and overall uniqueness for each of our cells. For cells to be different genes need to be read and turned into proteins in different cells, and to do this, our cells use gene expression. Certain genes get turned on or off, so some can be turned into proteins and some are just ignored. The specific combination of genes that are expressed or turned on determines the structure, function, and type of that cell. Once a cell has differentiated, it is impossible for it to become anything else. This means that the most special cells are those that have not yet decided, and these are called stem cells. We all began as stem cells, and so far these are all the ones that we've been discussing, specifically the embryonic ones that we began as. These cells are the ultimate wild cards of the human body because they can become any cell in our entire body. They have to because we all start as one of them, and we get all of our specialized cells from them. This power goes away as we develop, and soon the only stem cells left are the adult version. These cells are more limited, and they're only able to differentiate into several similar cell types. We know our genetics can determine specific aspects of our bodies, but how? We're going to use the example of achondroplasia, a type of dwarfism. Let's zoom into this achondroplasiatic person's 4th chromosome, the one that's responsible for this kind of dwarfism. The mutation on the FGFR3 gene that causes achondroplasia is almost completely dominant. This means that the parents and grandparents of an individual will always show dwarfism in the case that it is inherited. This is because the dominant version, or the allele, of a gene is present. The trait associated will always be present, no matter the genotype of its respective homologous chromosomes. Homologous chromosomes are chromosomes that code for the same trait in different ways. Think about when you hear 23 pairs of chromosomes. Each pair are homologous. Here we see 3-generation tree of inheritance, with the dominant achondroplasia allele present on one side of the family. We can see how the trait is visible wherever A is present, and the chances of the child inheriting the gene based on their parents' possible egg and sperm. Now that this achondroplasiatic person has their genetics, we need to examine how those genes actually correlate to a visible trait. The first step in this process is protein making. Here we see the DNA sequences that correspond to functional and non-functional proteins affecting bone structure and growth. Notice how just one difference in a codon set of 3 letters can completely change the function of the protein. mRNA is transcribed from the opposite strand of DNA, then shown here, changing the T's to U's but maintaining the fundamental differences. This information is sent to the ribosome where it is translated into amino acids and built into proteins. Here we see the effects of one letter difference from earlier. It changed one of the amino acids, which altered the structure of the protein, and which is actually the stem responsible for detecting growth. That stem no longer works. Now we understand how a trait is passed down through generations, and how those genetics affect what protein is produced. But how does this protein actually change our visible traits? The protein involved here is a regulatory protein, one that is responsible for bone growth. With the achondroplasia genotype, this protein loses its stem that signals for bones to grow, meaning it is always activated or sending a constant stop message. This leads to the inhibited skeletal development of the person with these genes, which is our visible trait, dwarfism. We can now see how the structure of this protein affects its functionality, and how that functionality corresponds to a visible trait. There are 4 main ways that we get genetic diversity. The first 2, crossing over and independent assortment, happen during meiosis, when our gametes, eggs or sperms, are getting made. The third happens when the egg and sperm come into contact. And the fourth, mutations are part of the larger picture of evolution, which is our next segment. Crossing over is when 2 homologous chromosomes trade their versions of the same gene. This means that no 2 alleles will always be on the same chromosome over many different generations of reproduction. During independent assortment, homologous chromosomes have 2 different ways of lining up to be pulled apart. With multiple sets, that means there are countless different random ways gametes can be created. Random fertilization is the chance that different genotype egg and sperm meet to make a zygote. In our example with achondroplasia, there are 4 different meeting opportunities, with 75% of them leading to achondroplasia. And finally, we have mutations, which is the chance that a certain part of an organism's DNA is randomly changed, maybe resulting in a new trait, maybe not. This type of genetic diversity is the backbone of our next segment, evolution. It's time to zoom way out of our bodies now, and into a larger physical scale and time scale. That's right, it's time for evolution. But more specifically, what about this concept tells us where we came from, and what makes us human compared to other animals? The one fundamental part of evolution is natural selection. Let's take the evolutionary example of resistance to the disease malaria. It always starts with a new allele, remember that's a specific version of a gene, emerging in the population. Most of the time, this is a mutation like we talked about at the end of part 2. So in our case, an allele emerged that allowed people with it to have resistance to malaria, a deadly disease. Next, there are 3 checkboxes that need to be filled in order for this gene version to be spread in a population. The first one we already covered. There needs to be variation in the population. We have that with our malaria disease mutation. Second, the allele needs to be able to be passed down through generations. If it isn't inheritable, then it can't spread over time. And finally, and most importantly, there needs to be what biologists call a fitness advantage associated with this allele. This isn't what we think of as fitness necessarily. Biological fitness has to do with the species' ability to survive long enough to reproduce and pass its genes down to its offspring. It is important to note that there could also be a fitness disadvantage. However, the trait wouldn't be widespread because members of the species with this allele would be more likely to die before reproduction instead of after reproduction. With malaria resistance, we can check these boxes. We have variation with the increased resistance allele, it's able to be passed down through generations, and most importantly, humans that have this allele are more likely to survive for longer periods of time, giving them a greater chance of reproduction and passing on their genes to more future generations. It is also important to identify the selective pressure, which is what the determining factor in the fitness advantage or disadvantage. In this case, the presence of malaria-ridden mosquitoes in African regions is our selective pressure. Next up, let's zoom out even more. Instead of just one species over many different generations, let's look at many species over an even greater time period. Here is a phylogenetic tree of humans and various different types of monkeys. Every time the tree branches, that represents a common ancestor. This is the most recent point in time when 2 different species are the same. With the example of humans compared to these different monkey types, we see that humans did not in fact evolve from any of the monkeys pictured. Instead, we evolved alongside these apes and evolved from a common ancestor. Alright, let's move on to humans specifically. Where on Earth did we come from and how do we know? Well, the answer is definitively Africa. Why? First, archaeologists have found bones of pre-modern humans there. Think Neanderthal and other slightly different versions of humans. Second, the first modern human bones have been found in Africa. This suggests that pre-modern humans branched and turned into the modern humans we are today. Aside from fossils, we can look at different ancestry's gene pools to give us a clue of where we originated. the African gene pool is by far the most diverse of any gene pool, with countless different alleles present. European and Asian gene pools have a lot less diversity. According to our story of us originating in Africa, this would make perfect sense, as only some of the population migrated away from the hearth, and as they did so, they left behind some versions of the genes only found in Africa. Continuing with our origins, we can see this tree with modern and pre-modern humans. While Denisovans, Neanderthals, and modern humans were all different species, we can see by the arrows between these branches that they could still reproduce with each other and therefore shared a large proportion of genetics. Contrary to these early humans, modern humans do not fit the branching tree model. Remember, when we use trees in evolution, it is representing different species. The differences in human genetics, however, are just not a great enough extent to warrant a tree model. Think back to those gene pools of Africa, Asia, and Europe. All different groups of humans share the vast majority of our genes. Even the small percentage of unique alleles that some populations of humans have are still rare alleles, meaning they're not shared by all of that population. Furthermore, non-concordance is a fundamental aspect of our genetics. All this means is that our alleles aren't biologically linked. Just because somebody is tall doesn't mean they have blue eyes, for example. Because of these reasons, a trellis shape is actually far more accurate for representing humans in an evolutionary standpoint. It shows how there is constant flow between different gene pools, and most importantly, it shows us how we are all so much more similar than it may seem. Alright, that was a lot. But to disprove the ways this science is being used for hate instead of good, it is important to acknowledge the fact that the past 10 minutes have been grossly oversimplified. There are countless more processes, components, and ideas that represent the most accurate model of the concepts discussed. Alright, back into bad genes. Is it possible that all the murderers coming into America share specific versions of a gene that make them predisposed to violent behavior? It does seem believable. If genes determine so much about our bodies, why wouldn't they determine our behaviors too? While there is evidence that psychopathy is linked to several genes, the science just does not line up. Less than 0.5% of people even qualify for the definition of clinical psychopathy. But more importantly, the allele for this trait is not specific to any outlying gene pools, meaning it is unscientific to say that any group of people are more likely to have the psychopath trait based purely on genetics. Once again, it is important to understand that the mental health issues that contribute to violent actions have to do much more with trauma and past experiences than genetics. Furthermore, we know from our trellis model of human genetics that any allele that specifically belongs to a gene pool is never present in all members of that population. This means it is incorrect and unjust to label all members of a specific gene pool in accordance to the chance that they have a given allele unique to their pool. With this in mind, we can conclude that President Trump was false in his claim that immigrants have a genetic predisposition for violent crime. The dangerous effects of this claim are apparent, as it is being used to justify harmful governmental actions. Not only are innocent people getting violently removed from the country based purely on where they come from, the idea of immigrants and crime have been closely tied together, using stories like these in the media, convincing the masses that these people are truly a danger to our society. That couldn't be further from the truth. Countless studies show that illegal immigrants and legal immigrants commit violent and nonviolent crimes at the same rate or lesser rates than native-born Americans. The data is clear, and so is our responsibility, to see people as individuals, not scapegoats. Let's choose understanding over ignorance and advocate for a future rooted in reality, not rhetoric.